can hear and see you.
All right, perfect.
Stage is yours.
Okay, this works as well, the full screen version?
Yeah.
Okay, good. Good afternoon, everybody. I'm Jonas. I'm a postdoc at the Niels Bohr Institute in Denmark. I will be briefly speaking a little bit about pattern formation in non-equilibrium systems, the information that is contained within these patterns, and the cost to maintain such patterns, basically. And I will specify a little bit within the realm of boundary-driven systems. So the motivation for these kind of studies is basically that we observe, of course, that in biology, animals have many different patterns, but why do they have these patterns, right? When an embryo is fertilized, it's basically just a clump of cells, which is almost symmetrical. So how does a barely fertilized embryo know which part of it should be a head, which part of it should be a tail, and whether or not it should have stripes, right? If there was an initial asymmetry, initial cells could maybe detect where in the anterior posterior axis, so that's the central axis of an embryo, where they are. But when it's symmetric, that's not the case. So the central question is basically, how do cells know, in a sense, what fate they should assume? So since the 70s, this is known that this is caused basically by chemical gradients called morphogens that are induced in stages, basically, during the embryogenesis. And an example I show here is a Drosophila embryo. So basically, that's the common fruit fly that you find on your bananas in the kitchen. And when it is fertilized, a couple of gradients arise at one end. It doesn't really matter which end, that diffuse throughout the system. And individual cells within that system can detect the local concentration of that gradient, and then based on some kind of thresholding mechanism, can initiate some downstream chemical reactions, which in turn, again, induce the release of other morphogens and so on and so forth, in the sense that at some point, basically all cells know what to do, know what to become, and how to react to different chemical gradients. So, for instance, if we look at this fruit fly embryo, and I boxed here on the left side this figure with the purple and the white, we have some gap gene that is expressed in a gradient-like fashion, which is called hunchback. And if we now detect this in experiments, we see basically that in an ensemble of different embryos, we find that the morphogen profile, or the gap gene profile in this sense, has some particular shape, but there are a lot of fluctuations, of course. We have thermal fluctuations such that particle concentrations are not identical in every embryo. So noise can actually destroy the ability of these cells to perceive basically the local concentration and react to it accordingly. So how would we measure this kind of information that is now contained within a concentration gradient and that's which cells can read out? So basically, if we pick one cell at random in the system and we measure a local concentration G, the information we gain from that measurement is the difference between the Shannon entropy of measuring and not measuring, right? So if we pick a cell at random, we have the Shannon, the uncertainty about the position is basically just the uncertainty in the distribution of cells in the system. But we gain a measure of information if we first measure at a specific position what the concentration there is, and then based on the conditional distribution, we can, with a relative degree of certainty, say where is this cell located. So because all of these concentration profiles, they get drawn as well from a distribution, we can average out this concentration itself, and we can construct an object that's the mutual information between position and local concentration. And due to the symmetry in the mutual information, which basically tells us that if we have information about the concentration, we get information about the position. It's the exact same thing as the information between position and concentration. So it's symmetric. We can basically change this formula into a difference between Shannon entropies of measuring local concentrations given a certain position, which is basically easier for calculations. So in our model, we assumed initial uniform spatial distributions, which is also something that's observed experimentally. So initially, cells are simply in a line close to each other, uniformly distributed in a cell, which is, of course, the reason why we would even need this kind of symmetry breaking through morphogen gradients. And as a side note, in the experiment I showed before, this specific morphogen or gap gene hunchback provides about 2 bits of information. And together with the other gap genes that are present in this system, the cells can actually know their own position within 1% accuracy. Good. So what we did in Copenhagen, in the group of Karel Proesmans, we basically wanted to study a simple toy model where we would have access to all the statistical moments, so we could characterize fluctuations as well. So we basically built a simple symmetric exclusion process with reaction kinetics in the bulk. So what this means is that we have particle exclusion, we have particle hopping on a lattice with an exclusion-based interaction and nothing else. They hop with symmetric rates, which fix the diffusive timescale, but they can also, at specific sites in the lattice, which I colored blue here, they can attach or detach in the system, which is basically representative for the downstream reaction that causes the next stages in the development of the embryo, for instance. You would have some kind of profile of, let's say, maybe enzymes that are localized in a system which can locally catalyze away some of the substrate or some of the particles in the system. So we have a combination of reaction versus diffusion, basically. Due to the exclusion interaction, basically we have a very simple model where we have a binary occupation variable, and we can then go to the hydrodynamic limits exactly. So in this limit, in the steady state, we can actually find that the dynamics of the system is described by second-order differential equation, which depends on 2 parameters, an alpha squared and a gamma. And this alpha squared is simply the ratio of the 2 timescales in the system, so reaction versus diffusion, while this gamma measures basically the direction of reaction, or the direction of this Langmuir kinetics in the bulk. Either particles are on average pushed out of the bulk or absorbed into the bulk, depending on the on gamma being smaller or equal to smaller or larger, sorry, than 1/2. And once we solve this differential equation and we know the density, we basically know everything. So our goal was now to study trade-offs between 3 different quantities. So first and foremost, of course, the positional information carried by the different profiles. The globally integrated Langmuir current, which is simply the current that the in and outflow current through the bulk, so not the boundary fluxes. And then finally, the entropy production, which is simply the thermodynamic cost to maintain that specific density profile in steady state. So if we look at it, we actually find that we can split our system into 2 different types of behavior, depending on when we see the maximum positional information. What I mean with this is basically, if we assume now, for simplicity, that we have a uniform Langmuir distribution, so throughout the entire bulk, we can have production and or decay, there is no spatial dependence. We can actually see that for some values of alpha and gamma, so illustrated in the top right here in this phase plot, we actually see that the maximum positional information can be reached either when the reservoir density difference between the 2 ends is 0 or 1 being the maximum in this case. So in the white region of the phase space on the top right, the maximum positional information is reached when the reservoir density difference is 1, and in the black region it's 0. But what this means in practice is basically, if we look at the different trade-offs, so for instance, if we look at the 2 middle plots, so the second column, the positional information versus a current, we can see basically that the highest positional information in the white region of phase space is always set by monotonic density profiles, which is the prevailing assumption in the literature currently about this positional information, that these kind of monotonic profiles carry the highest positional information. While if we now increase, for instance, the reaction rates as compared to the diffusive time scale, we actually find in the top row, in the middle one, we actually find that the non-monotonic profiles can now carry the highest positional information while still also carrying the highest and lowest currents. So there is a fundamental difference basically between these two kind of behaviors. And in my opinion, actually, the most interesting thing happens when we look at the trade-off, the Pareto optimal trade-off between the thermodynamic cost and the positional information. So if
You're
we
not
look at
cutting into your question time, just to be aware of that.
Sorry?
You're not cutting into your question time, just be aware of that.
Okay, yes, I'm almost finished. Sorry. Okay, so final thing then. If we look at the entropy production versus information, we see basically in the white region, we have a nicely monotonic trade-off between the two, where monotonic density profiles dominate everything. And if we now go to the black region of phase space, we actually see that we have a switch between monotonic profiles and non-monotonic ones, characterizing the highest degree of positional information. So there is some kind of phase transition in the optimal protocol when going alongside this Pareto front. So yeah, this is basically what I just said. So for both these plots, we have a law of diminishing returns. So if we want to have higher positional information, this of course leads to progressively higher dissipation in both cases. So for the future, we want to do some things like studying interacting particle models, or because, of course, this exclusion process is very easily extendable to asymmetric ones, we are also investigating this one, or optimizing the prior position profile with the bulk reaction profiles, because now we assume them to be uniform, but they can basically be anything. So yeah, I'm sorry I went a little bit over time, but thank you, and I'm ready for any questions you might have.